If you're ready, you can go ahead.
All right, let's see if this works. Is that on the screen? Can you see that?
Yeah, we got it.
All right, perfect. So thank you for the time. I really appreciate it. So what I was wanting to do, and like I said, I really appreciate the chance to recap 22 and share some of the items that what we've learned, and then what we've done to tweak it to fix some of the issues we had a year ago. And so I want to go through about 5 things that we have learned talking through our trials around the world, through talking to other people in the that are in the biological industry. And one of the things that, and there's no specific order of these, but one of the items that we've seen, and no one's really sure yet, but we all know that EDTA is not an environmentally friendly or a microbial friendly chelator. And we all know that zinc is part of a fungicidal and bactericide agents like in mancozeb and stuff. What we don't know is why sometimes when it's mixed in a tank with microbials, sometimes the microbials are degraded and then other times they're not. We don't know the reasoning behind it, but we've done a lot of research and we're finding that there's something going on there. And so we're going to take a closer look at that, and then we've got our own answer for that. And I know Monty's is working on one too, with different types of chelation. The second part is I didn't think much about this a year ago till started doing a lot of research. And every time we till the soil, we hurt the fungi, the mycorrhiza specifically, the trichodermas. But every time we do, we tear them apart, and then they got to start over and re-inoculate around the root of the plant. Tilling doesn't hurt the bacteria, which is interesting. But and then so we were trying to re-inoculate and put some mycorrhiza out there. The problem was the amount we put out versus what's needed based on starting over. We just weren't getting enough out. And one of the other questions was, the food sources, how often they're fed. The bacteria really love the sugars and all those type things. Fungi need plant extract sugars, and some of the other sugars can feed them for a little bit. But anyway, we were kind of fighting the culture of tilling, no tilling. And then, what type of EDTA zinc's in the tank mix. There was some of those type of things. The other thing we learned was when farmers are putting out a high level of phosphorus in their planting mix, that it's actually making, the best way to word it in a layman's terms, it makes the roots lazy. They don't produce the mycorrhiza and things naturally to go get nutrients because they don't need to. It's kind of like putting a plate of donuts in front of me, and then a steak is a mile away and I got to walk to it. I'd probably eat the donuts first because they're easy. You fill up, you get lazy, you get fat, but you don't get that strength in the muscle. And that's the way the roots are. They're taking in all this fertilizer that's right there for them. We're feeding them on a constant basis. Nothing wrong with that. It's just, it's going against what we were thinking we could do in the high yield situations with getting your fungi to grow. And it's something we learned just from getting back some of the tests, because the lazy roots don't develop mycorrhiza. They don't need to. There's food for them. They don't need to have nutrients transported back.
So Steve, I got a question for you on that.
Yeah.
So how much is too much phosphorus in the furrow to prevent the colonization of mycorrhizal fungi? I mean, we hear that a lot, that too much available phosphorus is going to prevent the colonization of mycorrhizal fungi, but I have never been able to find how much is too much. Is it a gallon? Is it 5 gallons? Is it the 50-50 ortho poly? Is it 100% ortho? Have you guys been able to come up with those type of answers?
Today, I don't have the answer, but now that I understand this, that is something our company is testing and going to test for in 23. We're going to try to run some lab tests and find out at what point does it start, going backwards, basically. It's a great question, and I've researched it a bunch in the last 3 months and have not been able to find an answer anywhere. But so and you know what? There's nothing wrong with putting phosphorus down. There's, the soils have plenty in there, but we got to find a way to mine it. And I think we've found a way that fits in with the cultural practices. And then the other thing that really I didn't understand, how often in a high yield situation that you guys go back out and you're putting the carbon, the food sources down, feeding the bacteria and whatever fungi is there, because you're doing it on a regular basis, every 2 to 3 weeks normally, from my understanding. And that's not a bad thing at all. It redevelops the colonies. It gets things going. It just may not be getting everything, because there's certain things that mycorrhiza does. And the 2 things that mycorrhiza is really, really good at is opening the pathway on calcium, zinc, and nitrogen. So we're working on another strategy that fits perfectly with what is in the soil. Oh, the last thing I forgot about here. We also found that some guys use growth regulators in their planting mix, which is nothing wrong with that either. But that is also kind of developing some lazy roots, because it's not making the roots do their own work with hormones and stuff and naturally doing these 3 things, because it's kind of just giving it that donut again, which is nothing wrong with that. It's something that I didn't personally realize and think about, but by doing all this test and working with you guys last year, I get to understand more where we got to go, what we got to do. And the other thing was, guys that are on a high yield program for numerous years, you've been feeding this, you're well over 300 bushel, or you're working your way that way. If it's, newer guys, the question that I have asked a bunch of people, at what point do you know when you've hit your peak on your property for the way we're farming it, the cultural practices, everything we're doing with the microbial makeup that's there, changing as much as we can change. But, at what point do we know we've hit the peak? I don't know that answer either, but that's some of the things that we're going to try to narrow down now that we have some information now that we can start working with. And the Baumakers test, once they start coming back in 2 to 3 weeks, makes it a lot easier to really dial in on the microbial part and the colonization of the soils. So the question was, we have a couple choices. One, farmers can change their cultural practices. They can rethink fertilizer programs. They can limit tilling. And I do know some people that add extra mycorrhiza, and I didn't realize that when we were getting these tests back that they were showing really good mycorrhiza. One of the reasons was they were adding in extra pounds per acre instead of ounces. They were spending a bunch of money, in other words. So my question is, is that worth it? I don't know that it is for the average farmers. Maybe it would be worth it if we take a couple acres and we start messing around by trying to, having a goal of where we want to get that mycorrhizal level to and see if it changes the fields that are already performing really well. But I do know that with the program we've got now for 23, we can help the farmers bring their low producing fields up to meet the other producing fields, because we're going to help get those fields alive with bacteria. And we're going to really use bacteria to start building the soils up. We're not going to try to force the fungi in there if there's nowhere for it to live, and we're not really doing the things it needs to live. The really cool part is that the bacteria, and I'll show you that in just a few minutes, the bacteria can do all of the same things the fungi does. But when biomakers started, they always talked about you wanted a 1-to-1 ratio, bacteria to fungi. Getting your way that way. Well, that's great if you're in a place that is just a lab, for instance. It's not real world farming. So I did get one set of results back last year of before and after, because one of the things I wanted to know was, are we changing the soils? Are we doing anything to help the soils going forward? Even though it may or may not show up in the yield, and last year was one of those years that yield was very sketchy. Even the higher yielding ones were not where they were the year before. So I wanted to know, and I had one guy, I think I've got two more, but I've got this one I was able to get through. And I just wanted to share it with you. The original sample in the spring versus after inoculating and running the MAC program, including the biologicals with it. This is what the soil looked like in August. And the yields were okay. They weren't great, but they weren't any, they weren't any worse. But we were hoping to see, big bumps. And last year was just a weird year to judge a yield, as you guys all know. But if you just look here, we started the year with 373 species. And by feeding and doing all the things they were doing with the program and adding in the EcoVam a year ago, we increased to 649. His bacteria went up to exponential numbers. So it went from, power of 8. Is that 8 million, 8 billion? Anyway, it increased a lot, because that's like a billion every time you add a number to the exponential part. Fungi did the same. So we did what we wanted to do. We increased his soil activity. He went from only ecto to endo and ecto. He put, I think, 1.5 pounds down of EcoVam, if I'm correct. The strange part is the diversity went from 1 to 73 to 1 to 1,300. And it's because we increased the bacteria quickly. And this is what we started seeing, that there's natural fungi that maybe that'll just come back on its own. Let's really work on the bacterias. His diversity went from, biodiversity went from low to high, functionality went from medium to high, and his resistance for diseases stayed the same. His hormones in the soil, the microbes in the soil, auxin went from low to high, gibberellin went from medium to very high, and cytokines went from low to high. So his stress adapters basically over average went from low to they were started at low and medium. It all ended up at medium to high. And I think one was very high. So we did see changes in that soil structure. So you look at his pathways now. Did we do any good on the pathways? Carbon was medium, still medium. Nitrogen, unfortunately, low and stayed low. Phosphorus was low. But on the biomaker scale, I don't have the numbers, but I wish I had the numbers to see if we increased. Because it probably went from, if the scale is 1 to 10 is low, he probably went from a low of 2 to up to maybe a 6 or 8 or something like that. He's still in the low category, but it improved. You can see potassium pathway went from low to medium. Iron was high, zinc stayed low. zinc, nitrogen, and calcium are almost always the same, and they are in his sample. I've seen it in hundreds of samples. We took but manganese went from medium to high, sulfur, copper went down a little bit. Not sure what that would cause, other than maybe just balancing out some of the microbes. Magnesium stayed high. And then the going over to the fertility part, his soil test, the numbers. So we can see that by utilizing the peptides and the whole program and getting the roots more active, he actually solubilized and moved through that plant a bunch more nutrient, because you'll see it here on the nitrate, ammonia, phosphorus. We solubilize more phosphorus, and that makes sense. His bacteria and his fungi both increased, and they're both solubilized, phosphorus solubilizers. On the potassium, they're transporters, so they move more potassium through the plant. Sulfur went up. Calcium went down because we solubilized a bunch of calcium that was there and used it in the plant. Whoops. Magnesium, iron. We actually solubilized more iron. He didn't need it, but we solubilized more iron, and that's from the fungi and the bacteria increases. Zinc, and I'm guessing we must have added zinc, a bunch of zinc too, because that's a huge increase. That's not normally from the soil. That tells me he added a bunch of zinc somewhere along the line, or the number's just not right. Manganese, we solubilized a bunch and used it. I'm sure, shouldn't say solubilized, we used a bunch of the manganese. Copper also doesn't make sense. So there's something weird about his copper and zinc numbers on here. And then once again, this is a one-time test. We pulled it. Maybe they had just put something down or sprayed something. Not sure. Boron and sodium. So basically, in looking over those tests, you can tell that they solubilized some stuff. They assimilated more nutrient. I wish I had tissue tests or sap samples to see the actual plant. And I'm going to see if he has those. I don't have them. But it's obvious to me when you look at that test, we are slowly changing the microbial populations to beneficials. Remember when we started all this, we talked about it being a 2 to 3 year program for everybody, depending where they were on the scale, and how long they've been with your guys' program. So we've tweaked a whole lot of stuff. We've took a bunch of cost out. We've realized that we got to simplify everything. 3-4 products is too much, too hard for guys to run the program, stay on the program. And so what we did is we've got a bacteria package. We've increased the amount of bacteria, and it's named Avio. That's going to replace Ecovam. Price is going to be a little bit lower than where Ecovam started last year. And then to make it very simple, the StemTide, it's a 2-step program, Avio and StemTide. If they want to use ZCat with a little bit of soil surfactant, if they're doing the Coulter side dressing and things like that, they can use that. It'll work a little better because it's got that soil surfactant in it. But we're going to keep the program extremely simple this year. And the specialty products, we have now got a Chelan zinc, which is chelated with the peptides, which I'll talk about. We have the ammonium sulfate with the peptides, N11S15, and then ZCat is there. Where ZCat did not mix really well in some cases with some of the brands of the ammonium sulfate a couple guys used. So they can use StemTide at a half rate in that place. Make it really simple. So now to get a little bit more into some of the more details.
Steve,
The issues, yeah,
before you get away from
Yeah.
that, one of the guys have asked, what soil tests do you suggest producers use to evaluate mycorrhiza concentration? Is there anything other than beet crop?
not that I have seen. There's only 3 in the world that do these microbial tests. One's in Spain, and the other one is owned by Wilbur Ellis. It's Trace Genomics,
Yeah,
and they don't break out the mycorrhiza like Biomakers does.
right.
One thing I've got, and I didn't have time to put it on here. I think they sent me the actual every organism that's in there, and I'm going through all those and having them send me everybody's organisms so we can look at all the bacteria and all the fungi and try to pull out the mycorrhiza. It may be in there, but under small amounts, or they're not recognizing it. I need to sit down with them now that we're into this part to find out what's going on. Why are we only seeing ecto in all these farm fields? I know they used to be woods, but you would think when you're taking it in the middle of the season, there'd still be endo. And then we get to like this guy's report and it showed the endo there, which is what the plant needs.
Right.
So right now, Biomakers is it. I do know that Lance out in Nebraska is going to be on board. He's delayed. He's not going to do it in March now. Last I talked to them, they're looking end of May, 1st of June. Something got held up. Imagine that with Biomakers, that something got slowed down or held up. But I can tell you, Waters, I've run 2 tests now recently, and Waters had them back in 2 weeks.
Okay.
So as long as they don't lose your test like they do Terry's, you'll have it
Right.
back in 2 weeks.
Right. The other question is, of all the products the guys used last year,
Yeah.
is any of that carried over in their soils this year?
Yes. The bacteria will, any fungi that's still alive, if they had a cover crop, it would go around the cover crop. There would still be spores available that would regerminate this spring, but the bacteria absolutely carries over year after year. That's the other thing. Anytime a guy's spraying fungicides, it hurts the fungi, but it doesn't hurt the bacteria. So one of the reasons we've looked at just did this test extensively around the world, trying to understand cultural practices and realizing that a lot of our programs are actually hurting the fungi in the soils. But yes, the spores could still be there, and naturally they should come back by feeding them and doing the right things that way. But the bacteria stays all the time.
Okay.
So Steve, going
Yes.
back to your fungi to bacteria ratio about 5-6 slides ago, you had one at a 1 to 73. I believe that was fungi to bacteria.
Yes.
And then halfway through the season or at the end of the season, it was 1 to almost 1,200 fungi to bacteria.
Yes.
So what we've been taught and what we've learned is that ideally you would want a 1-to-1 fungi to bacteria or a 2-to-1. But what you're saying is, are you rewriting what we should be thinking about, going from a 1 to 73 to a 1 to 1,300 is more beneficial to the soil and the whole biome of it to make other things, I guess you call it solubilized or available? I guess I'm just a little confused on that. That's a big jump.
It's very well, that one's a huge jump. But if you look at the 1 to the 10th power compared to 1 to the 8th power, that was a huge jump in bacteria.
And so if your bacteria is staying until the next year, wouldn't that ratio continue to be off?
Yes.
Or even higher?
Yes.
Bacteria
Yes.
to fungi? And biome is believing that this is more beneficial to have those type of ratios?
So the answer is no. In a wonderful, happy world, you want to be 1-to-1 as best as possible. because of the way we farm, the way we do things, we're not a permanent crop. We're always pulling, even in no-till, that's going to be better because you're not tilling it up. But every time we put something in the ground, we rip the fungi apart and kill it. And so it's trying to re-inoculate it every year. You would need to put down a pound, billions of spores of mycorrhiza, for instance, to get that in there. And the price to do that becomes pretty steep. So we have done research on certain bacteria, and I'll show you those bacteria in a second, that they do the same things the fungi does. And I think this is new thinking, but we think that we can help the farmers by increasing the bacteria amount and not trying to worry so much about the fungi, except for the good fungi that are fighting your diseases. But those are naturally in the soil, and that shows you that in the pathogens in your function or your resistance. Almost every time if a guy has a lot of certain type disease, he'll also have a very high resistance. So that's Mother Nature doing that, and that's usually fungi. But the other thing is there's lots of bacteria that also fight those diseases. They're antibiotic. They actually kill that fungi, the pathogens. So we're looking at it. Let's see if we can help the farmer rewrite history, I guess, but do something that's going to help them, that keeps it simple and easy for the farmer to be able to increase his yield and make his fields more viable, use more of the fertilizer that's tied up, but doing it by super packing the bacteria and letting the fungi be where Mother Nature takes it. Does that make any sense? It goes totally against the books that are written, but when I show you what these fungi do, what the bacteria will do, I think it'll make more sense.
So Steve, one other question says, Steve, won't the timing of the microbial tests change the results from spring to fall? Assume fall should always be much higher with biological activity.
And I absolutely 100% agree. I have challenged biomakers to do a weekly test because I personally believe that your pathways, when that plant needs nitrogen, it's going to send out exudates and sugars that's going to activate the microbes that provide the nitrogen. And when it needs zinc and when it needs these other nutrients, I really believe it changes weekly. So I love these tests for snapshots, but we can't live on them as far as that pathway, because I do believe if we did a test every week, we could probably gradually see those pathways change from what the plant sends out and exudates.
Right.
Once you get past a green plant and we start to go, where the plant's going dormant, I see those microbials start to go to sleep. Now they're still in the soil, but they're not as prevalent as where they were in the middle of the growing season. Mm-hmm. So I agree 100% with that question, that it changes all the time in the season. And that's what made last year so darn frustrating, because it was taking 4 months to get a test result back. You couldn't use it as a tool.
Right.
If I can use this as a tool this year for 2 weeks, that's going to help us understand the biology much, much better. Does that answer the question for him?
Yeah, I think so. Thanks.
Yeah, Steve, I 100% agree with you that the plant send signals out. I mean, the question is how do we make that signal stronger? That is my question.
Right,
what I'm
and
saying?
that's what I'm hoping when I show you what we came up with, that will do that. That is our number one goal, to send a stronger signal out from the plant. I agree 100%. We thought we had it last year, and we obviously didn't have what we thought we had. But we've learned a bunch through, and I appreciate you guys letting us work with you, because this is so different than the guys that are, fertilizing once or twice a year. It's a whole different world out there for those guys. And I think we can do something really cool, but it's going to be, going to have to really work. And I'm hoping this test will help us see if we can get a stronger signal from the plant. So really quick now, going into what affects your microorganisms in the soil. Obviously, we know drought can change it. They don't die, but they'll go dormant. Some die, some go dormant. Plowed soil is one of the, now that I understand this totally, plowed soil is really bad for fungi. Does not hurt bacteria at all, but fungi is a hypha of netting that has to stay together. It's a webbing. And when you plow, you rip it apart. So it has to start over. High temperatures, we know that certain fungi don't like, soil temperatures when they start getting really high. Lack of oxygen, too much rain, floods it. Sooner or later, some live, some go dormant, some die. Lack of nutrients, which is the food for the microorganisms. And then chemicals. We all know that fungicides kill fungi. And it's going to have an unintended consequence on other types of fungi. And then pH can affect it too. We don't normally have too much problem in the whole Midwest with pH. So we came up with Avio, and I want to show you what the role of microorganisms are in the soil. And you have denitrifying bacteria. You have nitrogen fixing bacteria like the legumes on soybeans and nodules. Then we have nitrogen fixing bacteria in the soil that helps get it ready for the plant to take. Then you have nitrifying bacteria, which you have that goes from ammonia to make ammonia to nitrates, and then nitrites to nitrates. All doing this so that it can make it available to the plant. And then we have decomposers to get rid of, things like thatch and all that, the bacteria, the fungi, aerobic, anaerobic. All of that goes together to make the nitrogen cycle for the plants. You guys pretty much know that, but I just wanted to show you that that's how important bacteria is to this whole picture. Go by this. So what we did is we supercharged the bacteria from our Ecovam to this Avio. Here's the bacteria that we have in it and what they do, specifically these 6. And then there's millions of other ones in our soil. But these 6 basically are going to help with oxygen production, phosphorus solubilization, root protections, and antibiotic, the liquid form is, and antioxidant activity. Is going to help that plant kind of kick in its SARS. Next one's going to solubilize phosphorus. Cytophore has to do with our iron uptake. It breaks down iron, makes it available to the plant. And then we have beneficial root. It's and then really strong, fast occupation. It fills the area quickly, grows, colonizes quickly. Then the Megatherium is phosphorus and nitrogen. Then this one here, plant growth promoting rhizobacteria. Most of these are part of that. They are formed all around that plant, and they're helping that plant grow, but also they're protecting against salt stress. There's a natural antibiotic that fights off certain diseases. Solubilizing more, another one solubilizes more phosphorus. Bacillus subtilis is the #1 fungicide bacteria on the market for sale. A lot of people sell it as an organic fungicide. It's in our product, and it's a great nitrogen fixer, great cytophore. Plant stress tolerance, gives a little bit of a biostimulant effect. And then pumice is nitrogen, phosphorus, and it helps reduce heavy metal in the soils, kind of protects the plant from those. You put all that into a package to go with Mother Nature's package. And now all of a sudden, and by the way, there are some harmful bacteria, everybody knows that, but the beneficial ones is what we're looking at. So now we're going to engage the soils, and it's basically, we want to observe an improvement in the yield and quality at the end. And in the past, we used to think it was fertilizers that did that. Everybody always thought it was the fertilizers, organic fertilizers, mineral fertilizers, whatever, low salt fertilizers. That's our key. That's what we thought. What we've now learning is that is part of it, but the healthy and living soils that break down all those nutrients, excuse me, are the whole key. So we need the plant growth promoting rhizobacteria to form around the roots and promote plant growth and development. We need it for water uptake and fight off competition from pathogens, and gives you that systemic resistance within the plant. This is all in the soils that helps the plant do all this naturally, sending those signals out that we believe will be stronger as we get the right pathogens in there. And here's the things these plant growth promoting bacteria do, rhizobacteria do. They fix nitrogen, and you guys know this, you've been adding it on soybeans. If you grow soybeans, you've been adding it for years. It's one of the things they add into soybeans. But you can see all the different things it does. Nitrogen, phosphorus, phytohormones, cytophores for the irons, reducing uptake of heavy metals, protects that plant, protects it from salt, decrease stress-induced ethylene production. And then the organic acid, that's your signal sending out the exudate, so that organic acid down there. So it helps control that and getting the right ones out there to get the right nutrients at the time the plant needs those nutrients. And antioxidant enzymes to help the plant fight off the stresses. So nitrogen fixing, there's a lot of nitrogen in the air. What I never really thought about was the fact that the bacteria in the soil can grab that nitrogen and make it available to the plant. Whereas like in soybeans, it uses the nodules to do so. Corn can do the same thing through the soils. And if you think about it, we get a burst of growth on corn and grass and different monocots. Every time we have a thunderstorm, you can literally sometimes see that growth in the green up. And that's from it taking in the atmospheric nitrogens. And because of the soils, that's what the microbes in the soils, that's what does that. Some of them are phosphorus that make the phosphorus, mineralize it, take organic P and turn it into something the plant can use. It's there in the soils. We know that. It just helps solubilize it. And we see that in some of the tests from start to finish, where we have more phosphorus than we put down, for instance. Iron, same way. Out west, they have tremendous amount of chlorotic, iron chlorosis in the soybeans. And yet their soils are loaded with iron. Well, it's just not available. So one of the things they do, if they can build their soils up with the right bacteria, they can actually get more of that to solubilize and possibly, cut back on the soybean chlorosis. But iron's important for photosynthesis in our corn just as much. Phytohormones, same way. Your auxins, gibberellins, cytokines have to do with cell division and growing the plant, setting the fruit. If we get the soils right with the right bacteria, which these are, that'll help the plant do it naturally. Fight off the antioxidant stresses, drought, air pollution, soils, salts, heavy metals, and so on. So you get your abiotic stresses and your biotic stresses. That's what these bacteria are helping the plants fight. You know, salinity, you got a picture here of a plant that was treated with a bacteria, and then one that wasn't, and on the left here. Here's some, it shows the roots and then the peptides. We also have the peptides in our product too. So on the left is the root hair promoting peptide that's in the Avio and in Stimtide. And it will help promote more roots. Over here on the right, you see no bacteria on the top, what it looks like, and bacteria in those started plants there. You get a strong biostimulant action from these bacterias. There's more in the form of the bacteria. It's more stable than the fungi. It's easier, better shelf life, easier to mix in a tank. The whole ball, it's still a soluble powder, but it doesn't have that temperature problem that your fungi will have, where at 85 degrees it starts dying. The bacteria doesn't have that problem. And then tolerance to abiotic stresses, we've talked about that. So here's some plants that were treated and untreated. This is cotton that had Avio on it on the right and didn't have it in furrow on the left. Here's some seeds that were treated in the Petri dish. You got your untreated on the right, treated on the left. That just helped the germination quicker, more building that root. There's a little bit of the peptide in here, which is a tremendous seed treatment. Here's 7 days after, well, they said after planting, but really after that, you can see a big difference in just how the vigor of the seed. Stimtide is the second part of the program, and that is our stress preventer that goes with the herbicides and fungicides. So that's how we started. That's what we thought this product, that's all we thought this product did. We've learned a lot more since then, but it's still number one use, and number one best thing it does is prevent stress when you have a herbicide and a fungicide going out at a pint per acre. And it stops the burn, it stops the stunting, it stops that root stunting. How does it work? Well, it's peptides extracted from a plant, and there's a difference out there between amino acids. There's amino acids that are extracted, and then you put them into a chain of 2 to 50 amino acids, and that becomes a peptide. Our product is 99% peptide, 1% free amino acid. Most of our competitors out there that talk about this stuff are animal-based. Ours is 100% vegetable, comes right from soybean meal, extracted out, and has 18 amino acids in it. The other ones have animal protein in them, and they do not do a single thing for the plant. When you find a product that's really dirt cheap, and it's probably either a sugar, a glutamate, or it's going to be an animal protein, a lot of animal protein in it. The plant doesn't like that, doesn't use the animal proteins at all. Our product is, you're talking about unlocking and signaling the plant. That's exactly what Stimtide does. It's like a key. You put it in there, the plant receptor, membrane receptor takes it, you unlock it, and all of a sudden now you got inside your plant cell, and you're literally signaling that plant when it needs to go get more root growth. It's gonna send out the exudates to get more root growth. When it needs to fill that grain, it's gonna send out the exudates to get the right nutrients to fill the grain. Now the nutrients have to be there, but once it does signal it. And we have 5 or 6 papers just on that nutrient efficiency. We just trialed it all year long on nutrient efficiency with this product and realized we're getting a 10 to 15% better nutrient absorption use in the plant than without it. And it can be on the fertilizer, it can be sprayed to the plant, like with your herbicide, and it still lasts, when you're putting your different fertilizers out. The lateral root hair promoting peptide is really cool because this, and I'll show you this video real quick. This is 20 in this, a tomato cutting. This is 7 days, 6 days. I thought it was 7. They tell me now it's 6. But either way, it's basically promoting these root hairs. That's why it's such a good product in a seed treatment. And it's in Avio. But you get this that much quicker. You'll see the amount of water they filled it up once in between. And that's one of the benefits that we get out of having the peptide to stemtide product. And it's in, like I say, it's in all of our products. And because of what it does in signaling the plant to go do the things it needs to do. Fights off stresses, as we talked about before. Here's a test we did measuring MDA from glyphosate. And they literally could test it and see that when the stemtide was in the tank, they got less stress on the plant with this test. And MDA is, and I can't pronounce it, it's malad something hide. Somebody, Joe, you probably could pronounce it better than me, but basically it's plant stress is what that is. So we spent all last year, 80 countries around the world testing for nitrogen assimilation is what we were testing for. What we learned was that we get in the plant and we help the plant signal when it needs certain enzymes. We activate the 7 enzymes that bring nitrogen into the plant. So you can see here, you go from NO3 to NO2. If there's microbes and stuff in the soil and you got your nitrate reductase there, it doesn't have to produce it. But if it's not there, it'll go get it and make it so it will actually activate that enzyme in the plant. The plant will then send it out and go solubilize those different forms of nitrogen. And you can see the chart here. It's very confusing, but the sugars are the glutamates. And that's part of the cycle. It can use all these different types of fertilizers this way, the nitrogens that are in the soil. It can use the sugar nitrogen to go into the plant. It can use a nitrate nitrogen to go in the plant. It can use an ammonia nitrogen to go in the plant. It will convert all of those into food source by activating the plant. And that's the one thing I could send anybody that wants the scientific paper, I can send it to you, showing you exactly how that works in the plant, signaling the plant. So now we've said, okay, but how much are we signaling? What are we doing? Are we making it any better? Here's the 7 enzymes. So when we foliar sprayed it, we activated the glutamate synthase and some of those on the left here. And on the right is the roots. This was drenched, and then it activated the enzymes in the leaves and in the roots both. When we did a spray, we activated other enzymes, but it activated both the root enzymes and the leaf enzymes. So it could take in all forms of nitrogen and convert them to energy. And not only did it convert nitrogen, this is the part that we started to realize and test for last year. The 0 is untreated, the 5.0 is a pint per acre. So you can see that in the plant, in the leaves, we went from 29.1 to 32.5 on the nitrogen, but we also went up on phosphorus, sulfur, your potash, calcium, magnesium. The only one that went down was sodium. And I believe 2 things. One, it flushed it out a little bit from the stress tolerance. But second, we probably improved the calcium magnesium ratio that would flush it out. So now we started realizing we are a nutrient assimilation when this is applied, not just a nitrogen assimilation like a lot of other companies have. So it talks about here the modes of action, nitrogen uptake, getting into the root or into the plant through larger root systems, which we're doing with the bacteria and all that growing those roots. Better transporters. The peptides are a great transporter to move this nutrient up through the plant to where it needs to go, the top of the leaves, all the way out into the grain. Then we can we regulate the uptake in the plant, the nitrates, and so on. Under low nitrogen supply, we upregulate the genes that needs to transport the glutamate synthase and some of those type things. Under high nitrogen conditions, we upregulate the nitrate reductases and some of those that are not normally regulated. So it works for both low nitrogen and high nitrogen in the plant. And this is all the plants sending those signals out that Kevin was talking about a couple minutes ago. Here's some corn in a leaf sap testing showing a 7% increase with a side dress here, 7.9% on the second one, and 11% on the third, and different rates of side dressing. It's kind of small on the bottom, but this was mixing the ZCAT product with it because that's the product we had developed for them to go with the nitrogen at the time. And that just shows on the leaf, not a big deal. Timing with herbicides, absolutely with the herbicide and the fungicide. If someone wants to use it for things like grapes and stuff, then it's at veraison and things like that. You can put it through fertigation. In corn, we're doing the 2 apps, herbicide and fungicide. This year, if it's easier for guys that are doing thiammonium sulfate, they could use a half rate of StimTide, not use ZCAT, not worry about the mixing order and all that, because we had a couple guys that had a little bit of trouble. This will work just as good. It'll keep the cost down for them, and then they only got to carry one product plus the other Monty's products, of course. Once again, tank mix 1 pint per acre, compatible with everything, almost every chemical on the market. There's a couple other things we do really well with: heat stress and drought stress. This is infrared picture under heat and drought stress. Here's some hail on the left. It recovered really well. So if you get a crop that gets hailed and it doesn't completely destroy it, you could spray it with the StimTide and it will help bring it back quickly. Here's some soybeans. And then I wanted to quickly just hit on what chelate is, how we learned this year to chelate with the peptide that is a friendly plant-based chelation for using with zinc. And I know Monty's is working on another form. We're actually working with them to see if there might be a joint type of chelation. But anyway, the peptides basically are used to chelate when we're making the StimTide. We actually introduce the zinc into it during the process. It encapsulates that zinc molecule and holds it and then takes it into the plant in a very easy form. So we've tested against EDTAs across the whole United States, and everywhere we go, we get as much or more zinc into the plant, whether it's in furrow. Most of the stuff we tested was in furrow, and it's been as good as EDDHA or EDTA. We did it with iron. Here's a picture of the iron product. We've got calcium, iron, and zinc are the 3 that are in a liquid form. Here's showing you the Keelan zinc when we had untreated EDTA, and then ours at the equivalent rate for the EDTA, and then a higher rate. And this was a zinc stored in the grain on corn. The reason we like this technology and we like, humic acid technologies and stuff is they're safe and friendly to the biologicals, so they can go in the same tank and we won't ever have to worry about, anything hurting the biological if you're putting them in the same tank to go in furrow, for instance. It's brand new technology. It's a brand new molecule that we introduced to the world last year. And I wanted to share it with you guys. And like I say, I know Monty's is working on some really cool technology because we're working with them. And by the way, the stem tide can be mixed in with the humic acid if you want to. And, you know, there's people out there that have an amino acid in with their humic. We haven't been able to get them to go in the same jug, but we can get them to go in the same tank with no problem. So the basic program that I was looking at for this year, very simple. It's Avio with your carbon, your sugar sources. Keep doing exactly what you guys have been doing. And then we'll come back with stem tide, with herbicide, with stem tide, with fungicide. And if they want to use ZCAT, and when they're doing the side dressing, or anytime you're putting nitrogen out, you can. You can also use 8 ounces of stem tide instead of the ZCAT. It'll be the same price per acre, about $5 an acre, and to get that nitrogen and nutrient assimilation into the plant. The specialty use products are below. We had some guys try the different ones last year. The N11 S15s and 1100 with 15% sulfur. That's beautiful at reproduction. If they're not using them, thiammonium sulfate, it's a liquid ammonium sulfate with peptides added. Very, very safe, good sulfur type. We had a few guys try it last year and like it. It seemed to work well on soybeans and corn. And that's kind of what we learned last year and where we're at. I'd love to hear any questions anybody has or ideas or thoughts. I know that, Craig, you brought up this thing that we're supposed to be 1-to-1. I don't know that we can get there unless we completely change everything we're doing in farming. So I think we can accomplish our goals this way. We're pretty sure, and my scientists are pretty sure, that we'll just supercharge the soil, but we'll supercharge it with bacteria that do exactly what fungi does. Any questions, thoughts?
So Steve, can you,
people are thinking.
Steve, can you send me pricing and updates and all that on these products?
Yes. Yes. And we're, oh, by the way, I wanted to mention also, and I need to talk to Joe and Dennis, and I want to run it by Kevin and Terry also. We want to do something for guys that helped us last year, learn so much. We want to do some type of loyalty program for you guys in the MAC group, and then offer something going forward for your new members that want to try some of the different biologicals. We don't know exactly how we're going to structure it, but we'd like to get your guys' opinion and input, because you know your group a whole lot better than anybody else does. So if that would be acceptable, in the next couple of days, I'd kind of like to get a hold of you and kind of get an idea what you like, what you think would be good for everybody.
Yeah, we can try that.
Steve, if a 9% EDTA zinc increases the zinc in my plant, how is it such a bad thing to have in your soil? You know, and the end goal is to get the nutrients in the plant. What makes this other chelation so much better with getting it in the plant?
So there's different schools of thought with EDTA. Zinc is good. There's nothing wrong with the zinc. The EDTA chelation, or EDHA, are synthetic man-made chelators. They stay in the soil. Now, you talk to the company that makes it, the actual chelator, and they'll tell you there's no harm, no anything. But if you talk to biological companies that run tests, millions of tests a year, you test the soils, the EDTA stays in the soil for some time until your microbes eat it up. It doesn't have a very short half-life. We think it's detrimental. There's something happening in the tank mix when we're going in furrow and we're putting certain microorganisms in that tank, like fungi. They get degraded in that tank. Now, we don't know if it's the zinc, because is EDTA, actually chelating that? Is it complexing? It's working to get it in the plant, and it's keeping it from tying up the soil. It's doing its job. This is just a natural way. If you had a humic acid chelator, it'd be the same thing. It's just a natural way of chelating the zinc to get it into the plant. The only thing that we do know is our peptides are absorbed into the plant very quickly with the roots or foliar, and the molecules inside the peptide are small enough that they get into the plant also and start working. As far as answering your question, going into the plant, either one works. It's anybody's choice. It's just a matter of whether you want a more natural product that's plant-based chelator, or if you want a man-made synthetic chelator. They both work efficiently. In all of our trials, we've gotten as much or more zinc into the plant as what an EDTA did. And we're trying to make sure it's priced like an EDTA so that farmers, and guys that use it don't have to pay more. But it's really just introducing something new to the farming community.
Yeah. Well, that's like the challenge that we have, is because at the end of the day, if it doesn't increase the yield, I don't want it to waste my time. But you have all these different products that are all organic or natural and want to go down that path. But how aren't these things increasing our yield by 20, 30, 40 bushel at a time? And we're getting these 1-2 bushel increases, but yet when we use synthetic fertilizer like nitrogen or phos or any of those, we get a much bigger response. There's a huge disconnect there that I struggle with using a lot of these things to give us the yield response that we want back. I tried mycorrhizal 2 years in a row.
Yeah.
I couldn't get any response out of any of it. I used the dry, I used the liquid, made sure that it, did what it needed to do. And the company, the creator of it, said, well, if you're over 40 part per million in phosphate, it ain't going to do anything for you. Well, 40 is not that high of a number,
Yeah.
So,
yeah. Well, I think
that's just where the struggle's at for me.
Yeah, I 100% agree. And that's a struggle that I learned this year. You have brought up, and one of the things that's happened is because of the way we do things, mycorrhiza doesn't colonize unless the plant sends out exudates to feed it. When we're doing a great job of fertilizing our plants, it doesn't have to send out any exudates. So that basically what's happening is that mycorrhiza is slowly dying off. Even though you inoculated it, it was there for the short period. And the same problem happened to us with our EcoVam. Now, if I took a farmer, which I do have in Nebraska, who doesn't use any, it's his choice, but he doesn't use very much phosphorus when he plants, he got 30 bushel more corn in year 3. He got 5 to 10 bushel in year 1, another 10 to 15 in year 2, and 35 bushel in year 3 of doing this program of adding, but he was adding way more extra mycorrhiza and then using EcoVam with the bacterias and then feeding the sugar, but he only fed the sugar 3 times. But his yield, okay, first of all, he'd never been on a program like the MAC program. He was a conventional farmer that they didn't do any of this stuff. He just started doing it, and that's when his yields came up. But my point earlier was, what is the point of saturation for each of us on our farms that we aren't going to get much higher, or unless we completely change our cultural practices and never tear up that mycorrhiza. We always have something planted in that field. It's almost impossible to do that. So the way we're looking at it now is, let's supercharge the soil with the bacteria that don't get killed off by what we do. I think mycorrhiza is the answer, but it's not the answer for this situation. Does that make sense?
But here's the problem. We've got ground we've been no-tilling forever, and it's laying right next to our other ground that we conventionally work. How's come the no-till? Like I'm not arguing with what you're saying, but the problem is our conventional ground is beating our no-till ground year in, year out. I mean, the last, I don't know, 10 years that we've been doing this, trying to push the yields, even the ground that we don't disturb, where we should have a higher colony of each bacteria and fungi, still cannot hang with the ground that we conventionally work. So I'm not contradicting what you're saying. We're just trying to figure out why is that? I mean, yes, it has a lot to do with the soil, but there's something else going on besides just the soil for that answer. I think that I don't know.
My complaint with no-till is I don't think I think you get too much compaction. I think at times they may get, it doesn't get the oxygen, because you're not breaking it up at all ever. And that's the sad part of this whole thing. I don't know if that we have the right answer, but we're hoping that we can, going this route, build up the soils for the guys that are doing a little bit of tilling, even if it's just one time over, to break it up to get more oxygen and stuff in the soil. But I think no-till gets so compacted that it's not the answer. I've never been a big no-till fan. I just see a lot of residue sit there. I don't know. It's just, I'm more would like to work it up a little bit. But from the mycorrhiza part of it, if we tested a no-till versus, and that no-till has to have a cover crop because mycorrhiza's got to live on roots. And I'm wondering if we're still dying off, even in no-till, because we don't have a cover crop there.
Yeah.
If that could be part of that. It's something, once again, we need to take more than one test a year. It'd be something we should, if we really want to know, we should probably take about 4 tests on the no-till and 4 tests on the conventional. And I'd be more than happy to do that because I'd love to know that answer.
Mm-hmm. Well, I mean, no offense to this big green push that's getting pushed, the regenerative ag or whatever, but
yeah,
bottom line is you still got to, we're farmers, we got to make money.
Yep.
And we're going to do whatever makes us the most money. And like I said, I understand what everybody's preaching about messing with the biology, but why are we still beating the no-till, with the cover crops every year? We're beating it on our farm. So
that's
I don't know.
Hey, Kevin,
Yeah.
one thing that we're finding is that when you take a soil that has decent fertility levels to begin with, and you have good microbiology present, for instance, like your soils, the addition of outside biology can often send a condition like yours backwards. And we're assuming or thinking it's because the natives in the soil are killing off whatever we're applying, because you already have decent levels. Where we're finding in the opposites is where some of this biology plays a factor. Does that make any sense, Steve?
Oh, I agree with that, but it's kind of like, where do you find the fine line at? Like I said, I 100% agree by applying phosphorus in, I think that's where we made a little yield bump several years ago when we pulled the phosphorus out of our in-furrow. But, not everybody has our levels. So, of fertility. So it's pretty tough to tell guys, to give them the right direction, say, well, yes, you need it. And no, you don't, because I do believe some guys with lower levels, I think there is a benefit in it. So, but I'm not yet even looking through some of the yield data through our MAC group and the tissue samples. Who in the hell knows what that level needs to be to where you add, phosphorus in furrow and where you don't. I mean, we're chasing our tail on that.
I agree 100%. I've been trying to find the answer to that for a long time. There's nobody out there that can tell you.
Yeah. I mean, like I said,
Other than that, too much is not good.
yeah, that we all will agree with. And I don't think, anybody that I know of in our group applies too much, but, in the end furrow that I've seen, but that's why farming is so interesting, because everybody don't farm the exact same ground, and that's what makes it hard, to understand 100% what's going on.
Well, Kevin, I think you hit on something earlier when you said this plant needs to send a stronger signal out to get the nutrients from the soil. And so stem tide on other crops, we'll spray it 4 and 5, 6 times through the season. We've never done it on corn because corn was never profitable enough to put a $9 application out 5 times. But maybe we do know that every time we hit it, we would get a boost in the signal strength going out, nutrients coming in. We'll see a boost of those nutrients coming back, even though we're only spraying the corn, the crop. But we've never tried it in corn like I do on all the other, on vegetables and everything else, where we're spraying it every 2 weeks. Once we get to reproduction, we spray every 2 weeks on most crops. Maybe, because I do know it's a strong signaler in sending those exudates out to go get that nutrient. Maybe
So
that's something.
You're saying that's a $9 application?
Yeah, at the full rate. For the biostimulant effect, to get the nutrient effect, we could do it for half that price. You could do 8 ounces, and every time we do an 8-ounce rate, once we've used it once, we can get a little bump every time. And we've actually seen it where we do it more often at 4 ounces. We get big nutrient intakes from the plant soil. Maybe that's an answer to look at, because after I saw your farm, you have excellent soils. Mm-hmm. That's you said it at the field day that I don't think the biology is going to help you much, and some of the other guys have been on the program a long time, because your biology is what it needs to be for your area. But other guys, it helps.
Yeah, I agree with that. But for, there's a lot of guys in our group that does have, that has had manure or running humics and sugar, that does have, their biology been running a good program for a lot of years. Yeah. Maybe that is something that, we're missing out, and I hope that Stem Tide can do it, because we're going to make several passes with it to see, if that is the missing link to gain more yield. Because I'll just flat out tell you, I mean, I'm about at wit's end how to gain more bushels. I mean, we're to the point now. I mean, I honestly feel like on our farm, we're almost to the point we don't know what else to even try. I mean,
yeah, because you got your timing. You've got your timing perfect now. When you got to get your nitrogen, your phosphorus, or your sulfur out, your program is dead on from everything I've ever seen.
So I mean, that might be something we may have to get, several guys in different locations across the U.S. or wherever across the seas too. We need to start getting data where applied on and multiple passes, in their playpen field, which everybody should be having that. But, to see the next big breakthrough on gaining yield and ROI, because like I said, I mean, there's only so much more out there a guy can do. And we're just to the point that, other than changing rates on sugars and humics, I think the fertility is where we need to be at. There's only a few other things that we can do unless we can get the plant to take up more, and that's what it would be. It would take something internally, I think, in the plant to say, hey, I want more of this. And
that's exactly what the peptides do. So as you were talking, it started making me think, that might be an answer. And you don't need we don't need the whole biostimulant effect the whole time. We just need the nutrient assimilation.
Yeah.
Which is between 6 and 8 ounces.
Okay. All right.
So that would make it a much more reasonable price for everybody too. Even at full retail, you guys get a better deal than that. But even at full retail, you're talking, $4 an acre.
Yeah.
But if you're limited on fertility and you're making that plant call for more, wouldn't that hurt it?
If your fertility's not there, yes, you can only grow what your fertility's got.
Yeah.
But what it will do, if you have it in the soil, there's enough carbon, and then the stem tide signals the plant to send out the exudates that's going to solubilize the nutrient the corn needs and bring it back in. So it's supposed to make the plant, this is the way it's always worked in all of our science, it signals the plant to go get the nutrients the plant needs at the time the plant needs it, because the plant brains know what it needs. If you don't have any phosphorus in your soil or don't have any nitrogen and there's nothing to convert, then we'd be in trouble. But if it's there and it's tied up, the way that the plant works, it would solubilize it and bring it back, because it's going to send those organic acids, the exudates, out to get that nutrient. And I think most of us have, for the most part, have a lot of nutrient in our soils.
But how does it know what it needs? I'm trying to dumb this down for a dumb farmer like myself. Like, how does it know what it needs? How does it know if it wants it for 400 bushel or 200 bushel?
The way I understand it from the scientists is the plant knows what it can produce because it's genetic. Okay, so let me back up. Let me make it easier. The genetics of the corn plant that you guys are planting has a maximum that they bred it for, whether it's 500 bushel or whatever that number is. The thing that reduces the yield is every time it gets stressed or it doesn't get a nutrient it needs at that time because it's not available or it's not getting transported up into the plant. The way these peptides work is they signal the plant, but they're also transporters, and they literally help that plant transport it up into the plant. So it goes out, the roots get it, and then it brings it back and transports it up into the plant. And supposedly, in all of our trials, we see that bump in our sap tests and our tissue tests and all of the science that we've got, that it moves it into that plant and where it needs to go. So if it's filling grain, it goes to the grain. If it's filling a grape, it goes to the grapes. And it increases the bricks. It brings more sugars into that fruit. Does the same with grain products. And the plant is smarter than all of us. It was bred to do its thing. We just got to help it signal to go get more of the fertilizer and then to be able to transport it back in. And one of the ways it gets more nitrogen is by activating those 7 enzymes. So the sends out these enzymes that break down the nitrate that's out tied up in the soil or the nitrites, and it converts them. It takes ammonia and converts it. It'll take that sugar you guys are spraying and help convert that into a food source. Instead of just maybe half of it goes in. We know that in our trials last year, we got 10 to 15% increase in nutrients uptake into the plant, out to the leaf and into the grain. So even if we can bump 10%, better nutrient efficiency, if you're at 80, 10% of that would be nice. If you're at 50, 10% is going to be a decent bump. And if we get to the high number of 15, then it would be better. It would be better yet. But the plant knows, the plant's smarter than me. I know it sounds silly, but they know what they got to do. As long as we're providing the tools for them to do it, and you guys are doing that with your nutrients, it's there. I believe it's there anyway. Does that
Yeah, I
make
agree.
any sense?
There's mud.
I can't answer the plant
can
how
give-
it knows.
If you can
We
give
need
me
to
more
find a breeder.
sunshine, that would be the most helpful. 'Cause at the
Yeah,
end of the
so-
day, that's a problem.
So sunshine.
Yeah.
So you just happen to remind me of one of the things. Early on, one of the things that we learned about StemTide was it helps improve chlorophyll in the plant, which increases photosynthesis. In Michigan, where I live, we see sun about half the time. In the winter, we don't hardly ever see it. And the crops that need that sun early in the year, when we've applied StemTide to them, they continue to grow like the sun was shining, and it increases photosynthesis. We've actually seen with one app through our, testing the the chlorophyll in the plant, it literally will keep the chlorophyll higher in the plant from day one on. Even with just one app, it'll stay higher than a control when you don't have any sunshine. So a little booster here and there. We've never tested it with boosters and stuff, but I wouldn't be a bit surprised that every time you hit it, you're going to get that little bit of kick in it. I know you get the kick with the nutrients. We've seen that. We've not tested the little boosters for photosynthesis and chlorophyll, but we knew one app stays with it the whole life of that plant in the growing season. We do have data showing that. So it helps
Steve,
offset no sun.
so Steve, before we wrap up, one thing I wanted to ask
Yes.
about the EDTA and your phosphorus and all of that kind of thing you mentioned at the beginning.
Yes.
Was any of your tests done by adding the humics and the sugars to that test to see if that didn't help buffer and increase efficiency and take up?
We did not, but that's one of the things we are doing this year, was to add those two to them. And one of the reasons we wanted to team up with Monty's was because you guys have a really good humic and carbon source. And we've done it with our sugars, but we've not done it with adding the humic to, the good carbon. So that's what we want to do this year, is
Right.
doing both and seeing how well that works together. Because we
Mhm.
believe there's a tremendous marriage there, synergies between those.
Right. Okay. Any other questions?
Yeah, and I just want to say there's nothing wrong with EDTA. This is just another way to get a natural thing that the plant will take in, and the plant loves the peptides. That's how it takes it in, and it goes right with it. That's all this type technology is. There's nothing wrong with any of the others.
Gotcha.
I didn't want somebody to think I was talking bad about the other stuff. But thank you guys so much. I appreciate the chance, and I appreciate everybody last year that worked with me, and I promise to make it this year. I'm going to make a trip. I'm going to make sure samples are pulled, and we're going to get data in season. If anybody has their last year tissue tests or SAP or whatever you did, and you also had an in-season biomakers test, I would really love to get my hands on that so I can put it all together. Because I'm trying to collect data and put a story together like I kind of showed you today. I'm trying to get more and more of that. So if somebody has it, that would be awesome.
Okay.
Hey, Steve, thanks for being on.
Yeah, thank you,
Flint.
Steve.
And Terry,
Yep.
you got to get a new hat.
Yeah,
I
That's what
agree
they-
100%.
See ya.
See you later,
guys
Steve.
down in Florida, actually.
Yep.
Are you guys done?
Bye.
Oh.
See you.